In a world brimming with complexity, few creatures embody harmony like the honeybee. With tireless precision, she dances from bloom to bloom, each motion guided by millennia upon millennia of instinct, each act in service to the whole. And then there are the beekeepers, watchful stewards of this ancient symbiosis. Part agri-scientist, part poet, they move along their hives with the efficiency of Moe, Larry, and Curly, tending to the bees' needs as best they can comprehend, and with the infrequency of a waterfall in the Sahara, sometimes running off, flapping and flailing like a penguin on a hot sidewalk. This is their journey.
Welcome to Bee Love Beekeeping, presented by our great friends and partners in beekeeping, Mann Lake. Here's the question of the day. Have we found a solution to Varroa mites? Can we see it in the near future? Today's interview features researchers from the University of California, Riverside, who have been studying a possible answer. And spoiler alert, it's not a new treatment, it's genetics. I think you'll find it super interesting, but first, and not to get confused with my previous statement, here's a story fresh out of Australia about what they're working on to knock down the mites. According to a study led by the University of the Sunshine Coast, researchers have identified components in the venoms of the Tasmanian cave spider and the giant Japanese funnel web spider that killed Varroa without harming the bees. Lead author, Associate Professor Volker Herzig, said, "We screened 50 venoms, mostly from spiders and scorpions, by applying them externally to the mites. We found more than 75% killed the mites within 24 hours. We selected 2 of the most potent spider venoms for further analysis. We isolated a specific component called a peptide from each of these 2 venoms and applied them to the bodies of Varroa mites. The peptides killed only the mites while the bees survived. These peptides are fully biodegradable, and our findings suggest they could be developed into a commercial, sustainable treatment for Varroa mite infestation in honeybees." The study also includes collaborators from the University of Queensland, the University of Oslo, Ghent University in Belgium, and the Swiss Bee Research Center Agroscope in Switzerland. The next steps would include testing the 2 peptides by applying them in mite-infested beehives to assess how well they work under real-world conditions. There's hope coming from all kinds of directions, so stay tuned. I'd like to welcome 2 really fun guests to the show today. We've got Genesis Chong and Owen Wagner coming to us from UC Riverside. How are you guys this afternoon?
Hi, Eric. Thank you for the invitation. It's really, really nice. The weather, it's getting very warm in here, but it's kind of funky. Last week was cold, and this week it's warm again, so...
I don't think your cold is the same as the cold where I live. I've been to Riverside. Does that mean it was like only 75 in the afternoon or something?
Pretty much, yeah.
But it gets really windy, and bees don't like it that much.
There's a good place to jump in. Why don't bees like wind?
Usually, I mean, this is anecdotally, I like to have the hive entrances facing away from where like the direction of prevailing wind is, because if that comes in through the hive entrance, that draft is going to chill the brood. It makes it hard for them to, maintain homeostasis within the colony, so.
I've never seen a study on this, and you guys are the smart ones. You're at the university, so I'm going to ask you this question. I expect a good answer. Okay, here we go. And that is, up to about what wind velocity can bees fly? Because if it's too hard of a wind, they just can't.
I have no idea.
I actually don't know off the top of my head either. So
All right. Topic for your next PhD.
To Google it, yeah. But that's a very interesting question. I never thought about it.
Today we're going to be talking about the research that's happening out there at UC Riverside. All about Varroa. Can I call them Varroa resistant bees? Is that going too far?
Yes and no. Because I do agree with you that they do show certain like phenotypic evidence that they are resistant. However, we still haven't like figured out the mechanisms. Like, okay, what's making them resistant towards the mice? Like, why they have like mite levels under the threshold and why they don't get that affected as other stuff of colonies like honeybees. But so far they have been showing like a certain type of resistance that we still don't are not completely sure how does that work. Like, yeah, we have some hints, and with this like that study we got like directions or on where to follow up with that.
Okay. Well, time out. We're getting just a little bit ahead of ourselves. I think it would make sense to give us just an overall summary of what the research was without using too technical of terms, okay?
Okay.
So, just an overall summary. Here's what we did, here's what we were trying to find out, and here's some of the things we've learned so far.
I'm the apiary manager, so like I was working in the field with like these research hives, but conclusions for the project, it's yours.
Basically, how it started, it was that we keep, like Cyber, that it's part of my lab, we keep a very close relationship with the beekeeping clubs. And from beekeepers, we kept like hearing that, oh yeah, like feral bees, somehow they show like a different behavior towards the mites, that they don't need to treat them as often. Their colonies, sometimes they don't get that harmed by the mites. So we also, in our apiary, we do evaluations like regularly every 3 weeks. So then we can have like an overview of the colonies. And what was surprisingly is that in a terms of 4 years, we were like analyzing the data that we took from the evaluations, and we found that there was a trend. We call it Californian bees because of the beekeepers. Like that's why they have like these Californian bees, because feral sometimes can sound kind of like these are Africanized bees, which they are, but they are hybrids. They have a certain Africanization degree. And if we compare that with the ones from Central America, Mexico, or South America, they do have like a smaller Africanization degree.
And just jumping in for a second too, I think an important distinction to make when we're talking about the language that we use with these types of bees and moving away from feral is that we're seeing a lot of beekeepers. I mean, we also work with these bees in a managed environment. So there's like a certain process of redomestication that's occurred. Like these bees are not feral. We are keeping them long-term in Langstroth hives, multi-generational colonies in the field that we have tight data that we've been tracking and recording for upwards of like 5 to 6 years.
So let me make sure that I understand that then. And maybe we just don't have the right terminology, but you're talking about bees that are not straight Italian or straight Carniolan or something like that. You've got bees that are, quote, Californian bees. And probably you could narrow that down to Southern Californian, I would think. And they have genetics from Africanized bees, I assume Italians, maybe some feral of some other things. So, in other words, they've hybridized themselves over time. And bees can do that pretty fast.
Yeah, and that's the beautiful part of these, because California, it's a hub for the pollination, and for the almond pollination, they get how many colonies? They get like more or less.
I don't know the exact count, but I mean, it's one of the largest pollination events in North America.
I think it's one of the largest pollination events in the whole world. 2 million colonies, roughly. It's huge.
Yeah, and the bees that get sent to almond pollination, those are commercial stocks that have been like purchased for like from a breeder, and these breeder usually they terms to like narrow down like traits like honey production, behavior of the colony, and just like performance in general. So those are like the genotype or like the stock that it's around in that area. So that always get to mate with different colonies around. So that creates like a type of like hybridization. So when we are looking like the ancestry of these bees, perhaps like, okay, what lines does these bees have in them? We can find type of European ancestry, Middle East ancestry. So they tend to be like very diverse. But we think that that's because of the environment that it's around, that the beekeepers keep like having like this stock of commercially colonies from breeders that in some way keep this diversity on the feral colonies that then we keep in our apiaries. I don't know if you...
So for your control groups, you had these, quote, California bees, and then you must have had some other bees that were non-California bees. What's the terminology we're using for those?
Well, for those one, we were using just like commercial colonies that we got from breeders, and then we keep them in a common apiary. So they have like in a common garden. Usually when we do, well, not usually, when we do like experiments and projects, all the colonies are in the same place. So then they get like the same resources around, same environment. And basically what we were comparing was like the number of mites that these colonies were having in a period of 4 years. At the end, were like 236 colony, I believe. But that total of colonies was the total colonies that were among like those 4 years. Some of them died, we got new ones. Some of them did survive like longer and they were able to be like multiple generations, but we didn't add like that detailed information into the article because for us, what we were interested in was, okay, let's see the number of the mites that they have and let's see how often we were treating these two groups. Because we were treating these groups, we don't have like an actual like survival in reflected in the article, because then, yeah.
So what did you find about these California bees? How did they deal with mites better or worse than your control group of bees?
They manage it spectacularly. If you have a commercial colony, right, and you don't treat for Varroa, I would expect like that colony to not make it to the end of the year if you establish it as like a package at the beginning of the season, like in spring. Even by summer, you're going to see the mite levels skyrocket. And then by fall, you're going to see the population of the bees plummet. A lot of that is, expressed through like deformed wing virus, which we almost never see in like this California group that we were examining in the project. And you also just don't see that type of colony collapse. You can have very large hives. I'm talking like 2 to 3 deep boxes full of bees going into like late fall and winter at that zero treatment, and you're not seeing really any negative effects that we associate with like a high Varroa presence in the colony.
I think the question I have then is, are they dealing with Varroa better or do they have less Varroa?
That's a good question, because we kind of like start from the baseline, just like finding or proving the difference that beekeepers have been seeing in the field, like observations that we have seen in the field. So we kind of try to test it. So in that article, we don't have any mechanism like, okay, the colonies do this and this is why they are able to survive better. However, from the field, we got, okay, Californian colonies, they tend to be barely treated against the mites, and often they do keep deinoseman counts under the threshold. There are some experiments that has been done here in the lab and in San Diego where they have found no relationship with hygienic behavior. So that was kind of another direction that we took. Okay, if there is no huge difference in hygienic behavior, what's going on? Why they are keeping their mite levels under threshold? So what we did was we were comparing larvae from these two stocks, and then we were using some plates as observational arena. In the first part, we use 4 different ages from those larvae, but from the 2 different stocks, and they were in the same plate. And in the middle of that plate, what we did was we put some mites in the middle. We released them at the same time, and then we record that for 2 hours. And every 15 minutes, we will quantify, or we will see like, okay, if a mite was in the area of one stock or the other one, a mite in the area of a specific age, which and was very similar or the same as what we know in the literature, that the mites larvae around 6 to 7 days old before they get capped. So then they can go into the brood cell and hide on the bottom of that cell until that larvae get capped and start the pupation process, where the mite will feed from that larvae and will start their reproductive cycle. It will start laying eggs.
Let me just make sure I understand. It looks like the difference in the 2 kinds of bees is happening in the larval stage of the bees. And is it that the Varroa is not as interested in the larva of the California bees, or is it some other mechanism?
From that experiment, we found that the mites seems to be less attractive to the Californian larvae because the reproductive cycle for the mite is so important. So finding the perfect host or the perfect larvae for them to reproduce, it's a must, that they need to do it right. And they communicate with chemical communication and stuff. So from those findings, we go different directions that we were able to explore.
Quick break to discuss what just might be on your mind right now. Come on, admit it. You're imagining the upcoming honey harvest. Because no matter where you live in the world, it's either here now or it will be in the near future. And Mann Lake is making it so easy to plan for. Click on over to Mann Lake's website for an amazing selection of extractors, filters, bottling supplies, uncapping knives, supplies for comb honey, everything, and I mean everything you'll need. But if you aren't there quite yet, and the honey flow is right around the corner, make sure you have extra supers on hand. You never can have too many or be too prepared. And right now, Mann Lake is offering podcast listeners $10 off your next order. Just enter code MLBLOVE10 at checkout. That's manlakeltd.com, discount code MLBLOVE10.
Perhaps it could be the larvae emit a different smell when it felt like the presence of the mite, or maybe the mites like them less because when they eat from them, they don't get the nutrients that they need to lay eggs, or all these type of theories. But what we did was we did an experiment where we had 8 colonies from commercial and 8 colonies from Californian ones. And what we did was, from deeps established colonies, we did splits and put them into nooks, and then they all start with the same. We tried to make them start with the same amount of workers, brood frames, food frames from the colony, and with queens. And then they all start from the same baseline. And then from them, we didn't treat them, we didn't give them any supplement or anything. Those colonies already came with their baseline of the mites, since we didn't treat them or anything, and because they were splits, they were just like carrying like the mites that they were having like in their deeps. We evaluate them for a period of 4 months. After that, we had infestation day, where we collect mites from sugar shakes rolls, and then we took 100 mites and we randomly assigned to 4 of the 8 colonies the mites, and the other 4 were kind of controls, because we wanted to hammer colonies to see what happened if I have like these 2 groups of colonies and then I'm inducing a high infestation of mites plus the mites that they already are dealing with. What will happen for us will be, okay, colonies will collapse because they also were in nukes, so they have like less place. What we found was really interesting, because after that induction of infestation, we were like, besides like the evaluations, we were inspecting brood frames, and we were opening cells with pupae that are dark eye. So by that time, the mite already have offspring within the cell, and we were quantifying. It's a mother mite, it got reproduction because have like offspring, it does have a male, or there is a mother mite and the pupa it's dark eye and she doesn't have offsprings. Yeah, we're still working on that article, but that's coming. But it's really interesting because so far we found that there is something going on between the brood and the mite, that somehow the mite, sometimes it's not able to reproduce.
Genesis, why do you think this is?
That's a really interesting question, because after that, we were looking into the larvae bee blood. We were looking into the hemolymph to try to see, okay, if there is maybe when they are in the presence of the mite, will they kind of release component in their body that then when the mite bite will make them not able to reproduce. But at the same time, we were also trying to get the chemical communication of those larvae in the presence of the mite and without mites. And we still are mastering that out to see, okay, maybe when they are in the presence of the mite, they do release like different chemical components that make them less attractive or something like that.
Owen, 2 sentences. Why do we think that it's different for these California bees?
Bottom line answer, based off of the data that we are looking at, that we're seeing, right? We know quantifiably that the mites are experiencing much lower rates of reproductive success in these California hives compared to their commercial counterparts, and that is all happening in the brood chamber. When we look at like the lab work that we did in the past with like the choice selection with the different larvae, and then just understanding like the mite reproductive cycle, there is a critical component that is happening in secret, in a sense, encapsulated away in the brood chamber. And like really what is going on there, we do not have a definitive answer for at the moment. We needed to even just get to this point. We started with that question of like, why are we just seeing this mite resistance in this population, right? And we need to also quantify, like, are we actually seeing that, or are we just looking for an exciting story? And you know, that's years of collecting that field data and then being like, okay, no, this is quantifiable, this is real, and then just working from there. It's been really exciting. It's been a journey for sure, and I think we're really on like the cusp of, I think, being able to figure it out too.
It is. It's super cool. And this is why I'm not a researcher, because I'm way too impatient. So I'm like, let's get to it here. So the cool thing is you've figured out where it's happening. It's in the brood chamber. And now it's a matter of why. Is it a pheromone smell? Is it, I don't know, do they taste bad? They don't have the same kind of nutrition that those commercial colonies did. It's got to be something along those lines. Once that kind of thing is figured out, then maybe something can be taken. Let's say it is a smell, that for some reason Varroa doesn't like the smell of these California bees. Okay, How do we get that smell and reproduce it and put it in hives all over the country? Or is it something else? And that's really cool, the thought that that could actually be something leading us in that direction. And I know that's jumping way too far ahead for this kind of conversation. But as beekeepers who are dealing with Varroa every day and can't stand it, and a lot of us that don't like to put chemicals in our hives if we can help it, but we have to, it'd be so cool to find some other way of dealing with this, whether it's genetic or a smell or a whatever the heck it is. I don't know. It would be unfair of me to ask you to speculate. I already have. But where do you think this is going to go from here?
Well, that's a good question because like that research, I was doing it for my PhD. And I remember that for my qualifying exam, one professor asked me, how do you see the Varroa mite problem in 5 years? And I growled like, oh, I will figure it out. I will solve that problem. So in 5 years, I don't think that there will be a problem. At the end, I ended up with more questions than answers. So I think that that's something very beautiful about like research and science because we start like from a broader context of an observation, as Owen was saying, but then slowly we were kind of like narrowing down depending of the results that we were getting, giving us that direction where to follow. So, oh my God, sorry, I forgot the question. What was the question?
Can I hold you to your 5-year timeline? You're going to have this whole thing solved in 5 years?
Yeah.
You can go to a bee convention at that point, and people will put you up on their shoulders and carry you around.
Yeah, but well.
And Owen will be throwing rose petals in front of the whole group. What do you think, Owen? Where's this all going?
I mean, obviously, it has really far-reaching practical applications. If we can really crack the code to like, what is this like defense mechanism that these California bees are employing? And especially if it can be replicated. Varroa is not the only stressor that bees are experiencing, but it's like if we could remove that from the equation, it would be huge for the industry as a whole, globally.
Yeah, it's not the only stressor, but it's really the biggest one right now. Yeah, there are plenty of others, and some of the others are there because of Varroa. A lot of the diseases are passed around, or the bees are weakened because of it. Or in a place like where I live, where in the winter there's going to be 3 feet of snow on the ground, and we're going to have below zero temperatures. If the bees are weakened, whether it is from a disease or Varroa or something else, that's the kind of stressor that means they're not going to make it through winter. Okay, on a little bit different note here, I've interviewed some other beekeepers from Southern California that have told me, in their opinion, all of the bee colonies in Southern California have Africanized genetics. Okay, they're hotter than bee colonies from other places in the country. That's probably why they assume that. They're also telling me things like, hey, it seems like the hottest colonies do the best controlling Varroa. Have you seen any kind of a link between behavior and this resistance that you've seen in your research?
Not necessarily like directly, because we tend to requeen or in a certain way to get rid of those very spicy and hot colonies, because it's a liability to have like that in where we keep our colonies, because there is like people working on citruses, chickens and stuff. So we don't want to have any incidents of these things. And also, I'm aware that some, or mostly the majority of beekeepers that keep them in their backyard or stuff, they also do like a type of filter. So, but it's really interesting. These colonies give a type of hope to kind of like to the problem. And we know that there are multiple populations around the world that has, in a certain way, has built either resistance or tolerance towards the mite. It's interesting how nature, in a certain way, after a while of these two, like the bee and the mite, by just being together, how nature, in a way, not favor it, but it's kind of like it's starting to build that ancestry.
It sounds like the direction you were going is saying that it's very likely that if humans were out of the picture, natural selection in these bees would eventually fix this problem anyway. Do you think that's the case?
In a way, yeah, it's kind of like nature has figured out something how to deal with this stressor. But at the same time, I know that the mite won't stay behind. At some point, also might figure out that new way of the bees to be resistant, and then there will be like that fallback. So there is this interesting theory in ecology that it's called red queen. That it's when, in this case, the bee and the mite running after each other and they don't catch it, but it's kind of this loop when one upgrade and become more resistant, then the other one find ways to overcome that, and it's like this constant loop. But it's really interesting because the Californian colonies that we have, it's also shaped by nature. It's kind of like all these stocks that beekeepers have around, and then the feral ones that have been here, they have been just like breeding among each other, and then they have like this diversity in their ancestry. I do think that diversity, it's a type of key because give them more tools to use in a way.
And then it's just an evolutionary race of who can
Yeah,
evolve faster, the bees or the mites,
yeah.
until the next stressor comes along. Okay, we're running out of time. There's one thing on the show that I give every guest an opportunity to talk about for a second, and that is the most wild and crazy beekeeping story that you've ever had. And that can be anything from just really unusual behavior in the bees to something embarrassing or painful. You get the idea. And I have a feeling, Owen, because you've been doing this longer and your hands are in the bees more than Genesis's are. Owen, I'm going to give you a second. If you can think of one or two, I'd love you to share one.
I guess my favorite bee story that I like to kind of pull out of my back pocket and share with people is like, I guess, a little bit unique based off where we are. We have the March Air Reserve Base over in Moreno Valley. So it's just like a quick city away. They have some civilian scientists on base that are doing natural science work, and they reached out to me to help facilitate colony removals on the base. They didn't want to just be spraying a bunch of poison around, which I thought was great. I would have really never thought that that was something they would have been that invested in.
Yeah, well, it's also illegal in California.
Yeah, yeah. But I got a call from them once pretty early in the day, and they were like, "This is super urgent. We know that you're really busy doing your research on campus, but if you could come out today, we have a swarm that moved into a plane out on like the runway, and it was slated like it needed to take off for some like important training."
Like a fighter jet or something?
Yeah, and they already had like all of their technician crews out there, but they were all too afraid. There's so many bees flying around. No one wanted to chance getting stung. So I was like, okay, I can clear my schedule. I can come out there. And I brought one of the grad students with me at the time, and we got out there and went through a bunch of security and checks to be able to even just get out onto the runway. And then they stopped us and they were like, okay, well now you need to go and pick out from the tread of like the work truck's tires every piece of gravel. We have a lot of gravel down in the field, because if any of the gravel gets out onto the tarmac, that can really mess up and damage the wheels of the planes. So we had to spend, I was just on my back, like just digging out all of the gravel that had gotten stuck. We finally get out there. I don't think they really knew what constituted a big swarm. It was very small, like a few hundred bees. It just brushed them off. I sprayed a little bit of soapy water around where the bees were. I told them, oh, it'll help wash away the smell, so they shouldn't come back and trouble you at all again. And that was it. It was so quick and short. But they had built it up like this was some like major critical issue. Like there were thousands and thousands of bees. But I always like to share that one.
Like national security is at risk here. And you have these men that have been through boot camp and been to war and all kinds of stuff, and come get our bees. They're scary. I love it. I love it. Is there a part 2 to that?
I mean, I've done some really fun work over there. There was another hive that had gotten up into a palm tree. But I mean, that was like a serious colony. There was a lot of comb, and they brought out a really nice ladder. And I had to scale all the way up to the top and just like cut it down. We made a little pulley system with some rope and a bucket so I could lower down each section of comb to the ground. I really enjoy doing colony removals when I have the time to. I don't have that much time at the moment anymore just because the research is always going to be the priority. But it does feel like a really valuable public service I can do. I don't want people out and about, that don't really know very much about bees to have a negative bee interaction just because there was a bee in a valve box that they happen to run by a lot and then they get chased or stung. I mean, bees are so important, and I think they are generally quite harmless. There's just usually something, unfortunately, that, can set them off. Like maybe someone just ran their lawnmower a little too long by where they happen to be, or just making too much noise or vibration, or there was some smell or odor that just set them off.
There's more research for you. Why do bees not like internal combustion engines? All right, last thought, 30 seconds, Genesis. Why do you love honeybees?
Why do I love honeybees? Well, I don't know. I started working with ants, and then I switched to honeybees, and I just got in love with bees. They always teach me so many new things, and they always surprise me too. I was a TA in the bees and beekeeping class, and it's really nice and rewarding to see the students when they start the class, the perception that they have about honeybees, and then seeing their perspective by the end of the class and see the switch completely. Maybe they start the class afraid of them, and then they leave loving bees, respect them. It's kind of like as they lose that fear of the bees. So I don't know. I feel that's really cool and nice.
That's great. All right. You two, thank you very much for your time, and keep up the good work in all this research. I can't wait to see where it goes next. And I'm going to say less than 5 years, Genesis, this whole thing is going to be solved.
Yeah, well, feel free to check like the webpage of the lab, because there is people doing really cool stuff. Like there is Sakshi, she's working with engineers and physics to try to figure out a way to quantify defensiveness on colonies with sensors. So there are like really cool stuff cooking around. Yeah.
Thanks again for joining us on Bee Love Beekeeping, presented by Mann Lake. Another big thank you goes to Vita Bee Health for their support. Vita's Varroa control range of products includes Apistan, Epigard, and now Varoxan, extended release oxalic acid strips. Hey, thanks a lot, guys. And if you haven't yet, please subscribe to and follow the show, tell your friends about it, and click on over to beelovebeekeeping.com to sign up for our free newsletter. If you have a guest suggestion or topic you'd like discussed on the show, shoot me an email, eric@beelovebeekeeping.com. And remember, if you're not just in it for the honey or the money, you're in it for the love. See you next week.